So a few labs ago we were looking at the macromolecules and we talked about the four classes of biological molecules: nucleic acids, proteins, carbohydrates, and lipids. And we focused on the proteins, carbs, and lipids, and for the most part ignored nucleic acids. Well, here we're going to dive a little bit deeper into those, focusing on DNA and RNA, the two major molecules from that category that we'll talk about in this class. We'll also get into a little bit of biotechnology and how we can use DNA for various aspects of human life. So let's introduce DNA, RNA, and biotechnology. DNA, short for deoxyribonucleic acid, is the genetic material our cells use and most other organisms also use. Ribonucleic acid, or RNA for short, is a simpler form of genetic material, and we use this as well when we are conducting genetic messages. We'll talk about both DNA and RNA and their roles in the cell. Biotechnology, as the name implies, with bio meaning life and technology meaning technology, is how we can manipulate biological things like DNA to benefit humankind. So let's talk about DNA. DNA has a very specific structure. If you remember from one of the previous chapters, we talked about nucleic acids having the nucleotide as their monomer or their subunit. So since DNA is a nucleic acid, its subunit is the nucleotide as well. And the nucleotide, we see many of them here in this image, is this piece I am circling here: a phosphate group, a sugar, and a nitrogen-containing base. So this here, these three pieces, is one single nucleotide. DNA is a double helix, meaning that we have this spiral-like shape, and the double part meaning we have two strands making up the molecule. One strand wrapping around on one side, and the other strand wrapping around the other. DNA and RNA both use bases, and it's the combination of these bases that tells us what information, what message we're transmitting. DNA uses four specific bases: adenine, thymine, guanine, and cytosine, shortened to A, T, C, and G. Not every single nucleic acid will use these exact same bases, but DNA is limited to these four specific ones. And these four specific bases pair in very particular ways. Remember, we're a double helix, right? One strand here, the other strand on the other side. And if we look at the pairs of bases we see here that are bound by hydrogen bonds shown in these dotted lines, we notice that they're pairing up in a specific way: G with C, T with A, C with G, A with T. So we notice that the T's and A's are always bound together and the C's and G's are always bound together. We call this complementary base pairing, where these bases will only bind with one other base. It's very specific. So you'll never see an A bound with a G. That would not work. We also have a specific sugar for DNA called deoxyribose, hence the name deoxyribonucleic acid. RNA is similar, but a simpler look at nucleic acids. It's a simpler molecule compared to DNA. Its unit is still the nucleotide, so we still have that phosphate, that sugar, and that base as our monomer or our main structure. But instead of a double strand, we are a single strand. So think of like DNA like a sandwich, whereas RNA would be like an open-faced sandwich or a piece of toast with jam on top, right? You don't have the other strand sandwiching in the bases. The bases are freely exposed to the outside. We also have four bases involved, four different bases: adenine, we have guanine, and cytosine. We saw those three in DNA, but instead of thymine, we're replacing it with uracil. So anywhere where there would be a T, we have replaced that now with a U. The sugar is similar, but is called ribose instead. It is a little bit different from the sugar that we saw in DNA, and that's where the RNA name comes from, ribonucleic acid. So let's compare and contrast DNA and RNA together. If we look at the images that we have here, here's our open single strand of RNA, here is our double strand of DNA. DNA, being that double-stranded, is going to use the A's, T's, C's, and G's for their bases, and deoxyribose for its sugar. RNA also uses A, C, and G, but replaces that T with a U, and instead of deoxyribose, uses ribose. There's also a difference in where we will find these molecules. DNA, since it's our genetic material, will stay inside the nucleus. It's very limited to where it can go. But RNA, since it's more of a messenger kind of molecule, can move around. We do find it in the nucleus alongside DNA, but we also find it in the cytoplasm outside of the nucleus. So how do we make more copies of DNA when a cell divides? When a cell divides, we have to be able to pass that genetic information on. That genetic information is our instruction manual. And if we're not able to copy that instruction manual and pass it to the new cells, then how will they know what to do? So we have to go through a replication process. DNA will unwind a bit. So we have an unwinding process that happens via an enzyme called helicase. The open area is called the replication fork, and that's where we can start replicating that DNA. We've unwound, we've pulled apart the two strands and left them exposed so that we can build the new strands on those old ones. We call that process semi-conservative replication because we're conserving one of the original strands, but we're building a new strand on the other side. The old strand is called the template strand, and the new strand is called the replicated strand. We can assemble these new nucleotides and new strands by using an enzyme called DNA polymerase. DNA telling us what we're working with, polymer tells us we're lengthening something, we're making something bigger, and A's tells us that this is an enzyme. So if you ever see A's, think enzymes. RNA has a similar enzyme for when we need to make RNA called RNA polymerase, which does a very similar job to DNA polymerase. It's just different of which molecule we're working on. So let's look at an image depicting DNA replication in progress. So here is our original double-stranded DNA molecule. We have our enzyme helicase unzipping and pulling apart the two strands, leaving one strand open and the other also open over here. We call this the replication fork. We have DNA polymerase, who is building the new strand using the old strand as a template. So we can use this old strand as a template because of that complementary base pairing. So if the old strand has an A, DNA polymerase can read that A and know that the other side has to be a T. If DNA polymerase reads a C on the old strand, it will place a G nucleotide with it. So we have instructions on how to build the new strand right there in the old strand. And we have a leading strand and a lagging strand. When it comes to replicating DNA, we can only move in one direction. And so one of those strands is going to build towards the replication fork. We call that a leading strand because there's no breaks. We can just go straight towards that replication fork easy. But the lagging strand is called a lagging strand because it has to build away from that replication fork. So instead of being one nice long continuous process of building the new strand, we have to do it in chunks instead. And that really brings us to the central dogma of biology, that DNA is going to lead us to create something like RNA through the process called transcription. RNA is then used through a process called translation to build a protein. So when we're looking at the direction of how information, genetic information flows, the central dogma of biology states that we go from DNA to RNA to protein. So I mentioned transcription and translation. What do we mean by that? How do we go from DNA to RNA to protein? We mentioned using DNA to make more DNA. That's great if we're going to be replicating our cells, but let's say we're just trying to live our daily cellular lives and we need one of the many proteins that we use on a daily basis. We need to be able to harness the information in DNA to create that protein. Transcription is the first part of that process. Transcription is what we see here up inside the nucleus. Transcription is where we are going to follow kind of a similar process to DNA replication. However, instead of making a new DNA strand, we're going to make an RNA strand instead. So we see that happening here. We've opened up the DNA molecule and we are reading that open strand and creating an RNA strand in response. So again, it's very similar, but wherever there would be a T, we're using U instead. We also don't need to use the entire DNA molecule to do this. When we go through transcription, we're only using a piece. Think of it like if you were trying to make dinner. You wouldn't copy down an entire cookbook's worth of recipes. You'd only copy down the recipe you wanted to use. So you can think of the same thing with transcription. When we make our RNA, we're only making RNA for the gene or for the piece of DNA that we want to look at, that we want to use. We're not going to use the entire molecule. Once we've copied that down in the form of RNA, specifically mRNA or messenger RNA, we need to edit that RNA strand. So we add things like a 5' cap and a poly A tail at the end, and any extra pieces called introns we can cut out. Think of it like if you were editing the recipe that you copied down. If you were making something like fettuccine alfredo and it called for peas to be added to it and you don't like peas, you might cross that off of your recipe list. Think of it like that with the exons and introns in the mRNA molecule. We cut out the introns, the P's, and leave the exons alone. Now we have an edited mRNA molecule that can now leave the nucleus. We call that entire process I just described transcription. Once we go into the cytoplasm as our mRNA molecule, we can begin the process of translation. Translation is like if you took that recipe card and started to actually cook with it. You started to assemble your ingredients, put things together, preheat your oven, so on and so forth. This is where we're actually going to build the protein we need. So mRNA, messenger RNA, is going to join up with our ribosome. And our ribosome is made up of protein as well as rRNA, or ribosomal RNA. The ribosome is going to be the factory where we can assemble things together, and the mRNA will serve as the instructions so that the ribosome knows what to bring in. We're bringing in our amino acids via tRNA, or transfer RNA. Transfer RNA is going to have what we call an anticodon on it, which goes with a codon. Remember the complementary base pairing we talked about with DNA? When we're looking at RNA, we have codons and anticodons. Codons are going to tell us what amino acid we need, and the anticodon is complementary to the codon. So if our codon, for example, is AUG, our anticodon will be UAC. They are complementary to each other based on the base pairing rules. We read the codon in the ribosome. The correct tRNA molecule with the complementary anticodon will match up, and it will bring along an amino acid that goes with that codon found on the mRNA. And it's through a process of reading one codon at a time and a tRNA molecule bringing the appropriate amino acid that we start to build what we call a polypeptide. Once we reach the end of our mRNA molecule, we've built our entire polypeptide, we then can dissociate from the ribosome. So now that we've gone through transcription and translation, which I do recommend that you spend extra time on, as both of those processes can be difficult to understand. Now let's look at what we can use DNA for. What can we do with DNA? DNA is found in every living substance and is actually pretty easy to extract. In your lab manual, you have a video link where you will watch a video on how to extract DNA in your own kitchen. There's also instructions in the manual that you can use in your own kitchen as well. But let's talk about a few key components that we need in order to extract DNA. Namely, something that has genetic material, something that has DNA, which can be almost any food that you have in your kitchen. Things like strawberries, things like wheat germ, things like oatmeal, whatever it is that you have on hand, as long as it was part of a living thing at one point. You need detergent to break down the membranes for the plasma membrane and the nuclear envelope. And one of the other really important pieces you need is really cold, either isopropyl alcohol or ethanol. So for those who are too young, who are under 21, you'll want to use isopropyl alcohol, which is rubbing alcohol. For those who are 21 or older, you could use a really good clear liquor like vodka to do this. I'm not necessarily saying you should do that, but it would be something that you could use if you don't have rubbing alcohol on hand. I know in today's world that might be a little bit difficult to come by. So either of those options are viable, but we need that because the DNA is going to dissipate or precipitate out into that alcohol. Make sure to follow the instructions very carefully when you do this on your own, because if you don't follow the instructions, you stir things too strongly or you pour the alcohol in too fast, you won't be able to see the DNA. So make sure to follow the instructions, and if you have kids, this might be a cool thing that you can do with your kids because it's a generally very, very safe experiment to do, especially if you're using rubbing alcohol and not something like vodka. But what do you do with the DNA once you pull it out? That's a fun trick that you can do in your kitchen, but why even bother? Well, there's plenty that we can do with DNA. We can use it for solving crimes, we can use it for paternity tests, we can use it to determine a species. There's a long list of things we can do. But having the DNA in just a snot-like looking blob isn't quite enough. Sometimes we only want to look at certain parts, and that's where gel electrophoresis comes in. Gel electrophoresis is a technology that allows us to separate out DNA fragments based on their size. So we have this setup over here. We have this container, this tank, a gel here in this blue color, sample wells that are little indentations in that gel, and then there's a buffer, a liquid, over this entire gel inside this tank. Now we would add our DNA to these little wells and then run a current through our tank. So we have a negative electrode over on this side and a positive electrode over here. Now DNA in general has a negative charge, so it's attracted to the positive charge on the other end of the tank. But because we have a gel there, things are going to move a little bit slowly through our tank, right? We're stuck inside that gel. If you are very, very large, it's hard to move through that gel. If you're very, very small though, it's a bit easier. So like I said, large fragments are going to move very slowly. So ones that have a lot of base pairs are going to be very slow. Whereas if you are teeny tiny, you can move through the gel much faster. We also add dye so that we can actually see these fragments. And like I mentioned, we can use this for forensics, paternity testing to determine if someone's father or mother, as well as determining if a species is the same species as another specimen. and so on and so forth. This is what the gel would look like after it is run and after it has been dyed. So we see that there's various bands that are these darkened regions. The ones that are further down here are going to be smaller fragments, and the ones that are found up here are larger fragments. DNA also gives us insight into a lot of conditions and disorders within human populations. DNA codes for pretty much everything that our body does, and when there's a mistake in that code, that can have really big consequences for us. So a change in DNA can cause various disorders. If we change the DNA, that can change the codon. If we change the codon, that can change which amino acid ends up in our protein. If we change the amino acids in the protein, we've fundamentally changed how that protein will work. Proteins are very sensitive. They have to have the right amino acid combination, and they have to be in the right shape. If you change up the amino acids and the order that they're in, you change the shape. If you change the shape, you change or destroy the function. We see that with sickle cell anemia. So here's our wild-type hemoglobin DNA, which just means the regular run-of-the-mill hemoglobin DNA strand. And we see that the DNA codes CTT, which leads our mRNA strand to be GAA. And this is the amino acid we get. But if we have a change in DNA, we've changed this middle T to an A, CAT. Our mRNA has now changed to GUA, which has changed our amino acid to valine instead. That one small change in amino acids changes the entire shape of the hemoglobin molecule. The hemoglobin molecule under normal circumstances helps carry oxygen around our body, but since the shape has changed, it can no longer carry that oxygen the way it would. It no longer functions properly, which means someone with sickle cell anemia would have a really hard time with their red blood cells and with moving oxygen around their circulatory system.